Hello lovelies, in this video we're going to be looking at measuring biodiversity for your GCSE biology. We're going to go through the different ways you can do it and then look at the different measures you can take of biodiversity. This is really important that we get this correct because it could come up in your exam.
Hi everyone. Okay, so we're going to look at how we can measure biodiversity. So ways of measuring biodiversity to kind of calculate it, estimate it, and look at how biodiversity changes in different habitats. So in order to study the biodiversity of an ecosystem, ecologists, that's the name of the type of scientists that look at interactions in ecosystems, we need to know the abundance, and abundance just means how many organisms there are, or the distribution, and that means where they are. So how are they distributed around the habitat? And we're looking in a specific area of habitat. So we're not looking at like the ecosystem of the whole world. We're looking at kind of a specific area of habitat in a specific ecosystem. So by measuring the number of species in an area, or where the species are in an area, then we are able to investigate how various biotic and abiotic factors are affecting the population numbers. Now remember, we've talked about this before, but biotic is living and abiotic is non-living. So we're thinking about how the distribution of one organism might affect another. That would be a biotic factor. If we're thinking about how temperature affects the distribution, or where some organisms are found in a habitat, or how many are found, then that would be a non-living factor. It's impossible to count the exact number of organisms in a large area, as it would take too long, and in some places there's just too many organisms to count. For example, think about a rainforest. So if we think about in a small area of rainforest, just simply how many different organisms there would be, and if you tried to count all of those organisms from tiny insects all the way up to massive trees in a small area, it would just be impossible. So we don't ever count the exact numbers or try and work out the exact numbers. What we do instead is we sample. So we count organisms in a small amount of the area, and then we use that small amount of the big area to estimate how many there would be of each organism in total in the whole area. And we're going to look at how we do that, and then look about how we estimate those population numbers. There's two main types of sampling: random and non-random, or systematic, and we're going to look at both of these today. Okay, so it doesn't really matter what sampling method you're using, whether you're doing random and non-random, but it needs to be both reliable, so that you can be reliant on your results as actual truth, or as actual kind of would be able to be done again by somebody else potentially, or be able to be actual representative of the area you're looking at. So we need our kind of sampling to be robust. So what do we mean by reliable? It means the sample needs to be large. If you just sampled a really tiny number, so you just put down a couple of quadrats, or you just looked at a really tiny area, then that's not going to be very reliable, because you would have got a very small snapshot of what is the real picture, and it's not good enough to then use it as an estimation for the whole area. So the larger the area that you're sampling, so a whole beach, a whole forest, the more small areas you need to sample or need to be counted in order to make it reliable for that whole area. Representative, the sample needs to represent the area. So it can't just be taken from one small part of the whole area in one place. It needs to be representative, so the sample has to have been randomly taken from across the whole expanse of the area that you are trying to estimate. So if you've got a whole field, you can't just measure one corner. You have to measure each corner, bits in the middle. You have to have been able to sample equally across that whole large space for your sample to be considered representative of the whole, so that when we're scaling up and using our sample to estimate the whole population in the whole area, we make sure that our results and that estimation is going to be accurate. So the sampling method and the way you're going to sample is going to depend on what organisms you're sampling. For plants and anything that moves very small, for example, here in my picture, I've got some rocks, and on these rocks will most likely be limpets amongst this seaweed. So there will be seaweed here, and that's obviously not going to move or go anywhere. And potentially inside my area, inside my Quadrat sample, there's going to be limpets. If you're not 100% sure what limpet is, it's kind of like a little shelled organism, and it lives on rocks, and it clamps down. It clamps down and sucks and attaches itself to the rock. And so they very rarely move. They will move very, very slowly, and they tend to only move around when they're grazing, which is normally when they're covered in water. So when the tide is down like this and you can see all of these exposed rocks and this area, there will probably be limpets there, and they're not going to move. So although they're an animal, they're not an animal that moves, or it's an animal that moves very slowly. You can also use a quadrat. If you're going to be looking at animals that can move around and you need to catch them, then you're going to need a different sampling method. But for the first couple of examples we're going to look at non-motile or non-moving organisms and plants. So we're going to use a quadrat. Now a quadrat looks like this. It's normally a square, sometimes wire, could be wooden frame. I've also got one here, so you can see the white square that's been laid down on the picture. It often has an internal grid, so it's got grid lines inside to mark out small areas inside, and we'll talk about why that is in a bit. You can get different sizes, from quite small ones to 1/4 metre square to all the way up to 1 metre square. So we're going to talk about how we can use this to sample in the two methods, and then we're going to talk about how we can sample animals that move. So first we're going to start with random sampling. Random sampling is a way of eliminating personal choice, which is what sometimes we also call bias, in the selection of a sample. Now, some of you may have been taught or may think that standing in a field and throwing a quadrat is random. It's not random, okay? Throwing a quadrat is not truly random, because the whole area will not be equally as likely to be sampled every time you throw, because it's going to depend on how far you can throw the quadrat. So that depends on how big the quadrat is, how good you are at throwing, and also the direction you're standing in when you throw. Throwing a quadrat is not truly random for all those reasons. The more quadrats you can use to sample an area, the more reliable it will be. Remember, we've said our sample's got to be large. So if we asked how to improve the reliability of something, we can always say we can lay more quadrats. So the randomness allows us to make sure our sample's representative, remember. The number of quadrats allows it to be more reliable. So this is the method we're going to use, and it's pretty much the same for any example of when you're going to randomly sample an area to find out how many organisms are there. First of all, we're going to divide the area into a grid, using measuring tapes or tape measures, same thing, placed at right angles to each other. So what we need to do, and I've got a sort of small example here, say this is my field of flowers, and I would lay my two tape measures at right angles to each other. So I've got here and here. I've laid them at right angles to each other to create a grid. And if they are a tape measure, then they will have measurements on. So they'll go up in, so you say you can go up in 10 centimetres, 20 centimetres, however many. You've got coordinates, which you've literally laid like an axis down of a graph, because you've got numbers going up the y-axis effectively and along the x-axis. You can pick numbers. So you need to decide where you're going to lay your quadrats, and it has to be random, remember? So we're going to use random numbers, which means we will generate them using a random number generator, or you could pick bits of paper with the numbers on out of a hat. It doesn't matter. But each time you're going to need to pick an X and a Y, and then that will be your coordinates. So it will tell you how far along the X-axis to go, and then how far on the Y-axis to go. So say we picked 8 and 20. So I go along the X-axis 8, and then I go up my Y-axis 20, make sure I'm in the right space, and this is where I'm going to lay my quadrat. And that's how I've laid it. I've chosen an area at random by picking two random numbers, and then I've used my grid to walk along 8, walk up 20, and lay my quadrat there. So once I've placed my quadrat down, I'm going to count the number of individual organisms, whichever organisms I'm looking at, or I could estimate percentage cover. So now percentage cover is a bit more subjective, because what you're doing is you are estimating. So you're estimating the percentage of your quadrat that's covered by an organism. So if, say, 50% of my quadrat, in 50% of my quadrat squares, or half of my quadrat squares, I could see a daisy, I could say that roughly percentage cover of daisies in my quadrat was 50%. But that's very subjective. It depends on how you're deciding this, and it's not as reliable. Count the numbers of each species. Now, we could just be looking at one species. In this example, I could just be wanting to count yellow flowers. So if I just want to count yellow flowers, then that's fairly easy. I'm only going to be looking at that species. I know that that's the yellow flower I'm going to be looking at. But I could be wanting to look at kind of the relationships between different organisms, or looking at the different numbers of, say, yellow flowers to purple flowers in this field. but I might need to be able to identify those plants, especially if they haven't all got flowers on. Or if I don't know, I've gone to a field and I've said, oh, I need to count the number of this species and I don't know what it looks like. So I can use a key to identify each species, which is normally a little book, a little diagram that tells you features or characteristics to look out for to identify which species is which. So I make sure when I'm counting, I'm actually counting all the species, the individuals of that species in that quadrat. Then I'm going to write my number in a table. So I count the number of species of yellow flowers or purple flowers or both in my quadrat, and then I write down those numbers in my table. And then I'm going to repeat it. So I would repeat it again, get 2 other random coordinates, go somewhere else, put the quadrat down. So say this time we went over here, I'd put a quadrat there, then say we went here, I'd put a quadrat there based on the numbers that I've been generating, and then there's one here. And I just keep repeating that until I've considered that I've got enough of a representative sample, reliable sample, sorry. So let's see how many I can do. And let's say, we'll talk about in an example in a minute how many I've done. But once you feel like you've covered enough of the area and you've done a large enough sample, then you can stop. Then it's about calculating the mean. Remember, the mean is just another way of saying average number of each individuals of each species per quadrat. So for all of the quadrats, I add up the number of flowers that were yellow flowers, say, that were in each quadrat, and then divide them by the number of quadrats. Same, I could count up the number of purple flowers, the total purple flowers that were in all of my quadrats, and then divide it by the number of quadrats. Let's think about just checking how we count within a quadrat. So say this is one of my quadrats that I've put down. So say it's this one over here, and I've laid it down, and this is what I'm looking at. Obviously there'd be grass, there would be purple flowers in there as well, but just for a sake, let's have a quick look at how this would work. You have to decide how you're going to record the numbers of flowers in this quadrat. So what we tend to do is we won't count anything that's not completely in the quadrat. So these 2 here, I would not count because they are not totally within the square of my quadrat. So anything that's over the boundary does not count. So I'm going to have 1, 2, 3, 4, 5, 6. So the number of flowers in this quadrat is 6. And I would make sure I can keep doing that for all of my quadrats that I'm going to take. And then, as I said, so we would add up the total number. So add up the total number of flowers from each quadrat, divide it by the number of quadrats, and that gives my mean number of flowers per quadrat. If there were any extreme outliers, we tend not to ignore outliers in ecology because anything can happen. You're doing totally random distribution, so there shouldn't really be any major outliers. But if there really, really was something that you thought was really strange, like you accidentally dropped it on a path or something like that, and you're not looking for that kind of change in environment, then potentially you can ignore outliers when calculating mean. But because this isn't a reaction where we're predicting a result and we know what should happen, i.e. as X increases, Y increases, this is random, it's nature. It's going to be different, the distribution is going to be odd, we can't control it, so we shouldn't really be removing any outliers. So once we've calculated our average number of each species per quadrat, so just looking at yellow flowers, or you could be doing the number of yellow and purple flowers, whatever you're doing, we can scale up our sample to estimate how many there are in the entire large area of the habitat. So, for example, if our flowers in the field, then that area, imagine the total area we sampled of that field was 50 metres squared. So in the whole field is 50 metres squared. In total, we put down 20 half-metre square quadrats, and they were randomly placed across the area. In total, the total area we sampled was actually 10 metres squared, which is about a fifth of the entire area, which is probably fine and fairly reliable. My average ended up being that there were about 8 flowers per quadrat. So what we can do is to estimate the number of yellow flowers in the whole field. I'm going to take my average number of flowers per quadrat, which was 8, and then I'm going to times it by the total area of the field, which was 50, divided by what we measured, the sampled area of the field, which was 10, which gives me 40. So my estimate of yellow flowers in the whole field is 40. And that works for whatever you're doing, for any maths question that you're doing. If you've got the average number per quadrat, you've got the total area that was being potentially sampled, and then you've got the area of the number of quadrats that you specifically sampled, you can work out the estimation of the number of individuals per species. Okay, so non-random sampling. So a transect is a way that scientists can look at the distribution, remember that's where organisms are, of a species and how it changes as the habitat changes. It's useful for investigating how biotic or abiotic factors affect population numbers. Now, random sampling, you can do that. You could sample a field that has been mown and a field that has not been mown. Or you could sample a bit of desert that has really high temperatures at some point, and then you could sample a bit of desert or similar dry environment with low temperatures. And then you can just compare the numbers. What we're talking about here is about change within one habitat. So we still use a Quadrat, but we're placing it at regular intervals along a single tape measure this time. So we're not using a grid. And that single tape measure goes through a changing part of the habitat. And because we're placing the Quadrat at regular intervals along the tape measure, we're not randomly placing it. Therefore, it's systematic sampling. Okay, so this is the method we're going to use. So we're going to take a tape measure and we're going to lay it across the area. Now it should be going across an area of change. So I've got a couple of examples. We could be doing it from right underneath a shady tree to far away from the shady tree. So in this example, we'd be looking at changing light levels, and that would be like our abiotic factor that we think is going to affect it. We could also be looking at changing or how something changes as it gets closer to or further away from a river. So in this case, we'd be looking at water concentration, or moisture levels. So that soil is going to get wetter and wetter as we get closer to the water. And so we could be looking at how the plant's distribution changes as we get closer to it. So we know the habitat's changing along our transect, and we're trying to look at how that's affecting how many of each species are going to be there. We need to always start with placing a Quadrat at 0 meters, so at the very, very start. So it depends on what direction you're going in, whether you're going towards or away from. But for example, 0 meters could be here by the bottom of the tree, it could be here by the start of the lake. Place your Quadrat down, do exactly the same. So given what we've just talked about, 3, 4, 5, exactly the same as what we've just previously looked at, how we count species in the Quadrat. And then you're going to move the Quadrat along the tape at a set distance. So you will decide how far along you're going to put your tape. Are you going to put it every 2 meters, 10 meters, 20 meters, depends on how long the transect is. You will decide where to put it. So say we did it at 0 meters from the tree, then 5 meters, then 10 meters. It needs to be the same. Say for the river, we did it slightly differently. So we did 0 meters, then we did 2 meters, then we did 4 meters. As long as it's regular intervals, so every 2, every 5, every 10 meters, it doesn't matter, but the intervals need to be regular. So remember, this is not random. You then repeat exactly what you've done every single time you do a Quadrat all along the transect. And then this is the thing that is very important that people often forget. In this case, we can't just add more Quadrats. What we need to do is we need to repeat the transect. So it's got to be a slightly different position so the Quadrats aren't overlapping, but you need to repeat it so you're getting the same thing. So again, at 0 meters from the lake, again at 2 meters from the lake. River, again at 4 meters from the river. So each time what I've done is I've placed another transect, so I'm getting similar data, but the Quadrats aren't overlapping each other, so I'm not getting the same data, but it's still in the same area, it's still in the same kind of direction, and the distances will be the same. And that's how we're going to get our average. So we will average together the 0 meter Quadrats, we will average together the 2 meter Quadrats and the 4 meter Quadrats. Same for here, we'd average the 0s, we'd average all the 5s, and we'd average all the 10 meters. And then that would give us our more reliable sample by doing it more than once. Okay, so that's how you would make it more reliable. You'd repeat the transect in a similar direction or in a similar area. So what we're looking at with the transect, what we're looking at is a gradual change. So like we said, light, for example, or water, or it could be pH, it could be a lot of things. So a gradual change in the distribution of a species is what we're looking at, and that's called zonation. So how a species changes within a habitat. And it can be caused because of a gradual change in abiotic factor, which we've already given some examples of. We can use a kite diagram or a bar chart from the data collected to identify the changes in my distribution. So let's say, for example, I've got a different situation where I've taken a transect across a path. So I've got the left-hand side of my path, and I've got my right-hand side of the path, and then I've got the path itself. And you can see I've got my transect, and I've got my Quadrats, and let's say I space these out every half meter. So I've got 0, 0.5, 1, 1.5, 2, 2.5, and 3. So if I was going to look at the distribution of flowers, so let's say we'd be looking at, just say the different species of flowers across the transect. What I would do is I could plot that on a bar chart. So I'd have what was my transect along the bottom. Say I had obviously really heavily planted flowery borders either side of the path. Then I'd expect the early quadrats, I'd expect to find quite a lot of different species. So I'd have maybe between 6 and 7 species. So I put my initial reading here. And then let's say when I get to 1, I've got slightly fewer because I'm getting close to the edge of the path, but that's because I'm going to have slightly less space to plant loads and there won't be as many weeds and things. And then obviously when I get to 1.5 and 2, I've literally, that's the path. There are no species there. So then I've got some species, but remember that quadrat is sort of half on, half off the path. So it's going to have less. And then I'm back to having quite a few species again. So if you had not seen my transect or what I'd done, and you just had this data, this bar graph in front of you, how would we be able to tell where the path was? Seems like an easy question. So how can we tell from the graph where the path is? Well, it's where there are no species. So the path is clearly here. So I would expect the path to start around here and end around here. And now this is a question they could ask you. So this could be a river, for example. It could be a path. It could be that there is a bit of pollution or a pipe or something running through that's causing this difference in species. It could be they'd have done this with 2 different types of species. So there is an area where one type of species is growing and it's out-competing the other. So therefore you have a gap in distribution. So anything where we can see and use and tell the bar graph, we can use it to kind of give an indication of what the zonation is of species, how they're distributed along this changing environment, and then have an indication of it. We could, for example, do the same thing, but instead we may have a graph where, say, we've got, so the river is here, and we've been looking at one species in particular, and say this one species is adapted to living where there's a lot of water. What we'd expect to see is a trend like this. So as we get closer to the river, the number of that species increases. Equally, we could have one where we have the tree, and we could be looking at a species that can't survive in shade. So as you get closer to the tree, it's going to get more shady. So we could say, see a distribution that looks like that. So you can show, using these graphs, the distribution of the species and then talk about how those abiotic factors are affecting the distribution. So now let's think about if we're trying to look at the distribution of a species or the abundance, how much of a species there is in a habitat, but it moves around. We're going to have to be able to catch them. So we have to use specific equipment in order to catch them so that we can then start counting them. There's lots of methods and it's going to depend on what you're catching and where you're catching it. Does it live in the water? Does it live in the trees? Is it on the ground? Is it going to live in grass? And how big is the organism? What type of organism? Is it going to fly? Is it crawling? Is it big? Will it be attracted by food? All sorts. But you need to think about the fact that you've got to do it ethically, so we're not trying to harm them or stress the animals too much. And so we've got some examples. First one is a pooter. A funny name, I know. It's used to collect some small insects by sucking them into a little chamber. So it looks a bit odd, but the way it kind of works is you have a little kind of canister, often with a lid, and it will have 2 tubes coming off of it. And there will be a little filter here on one of these tubes. So what you do is you suck, using your mouth, you literally suck like a straw on one end, and that creates a little vacuum in here. And so then you put this close to a little insect, a little bug, and what will happen is they will get sucked into this little chamber, and then they won't be able to climb up and crawl out. So in an area, you've got a quadrat, say, or you're looking at a little area and you need to collect some, you will be sampling an area, so you'll be kind of sucking all the little insects you see, and then you can count them in here, and then you can take the lid off once you're done and free them. A pitfall trap is a small trap where you can dig a small hole, and it's used for kind of very small organisms. Could be kind of crawly insects that would be maybe too big to be sucked up into a pooter, for example, or you want to be able to leave the trap over time and come back to it. And that's going to be useful when we're going to talk about how we calculate this. So it can just be simple as you dug a little hole, you have covered it with leaves or something that will easily fall into the hole, so that a bug will walk along and the weight of it will cause it to fall into there. But this is deep enough that they can't climb back out again. And that's all a pitfall trap is. Sweep nets. So these are just really large nets, often with very, very fine mesh. So something that you can, so they normally have really long kind of area on them, and then a big kind of area that you can then drag across, say, long grass. Often they can have tall handles so that you can use them in the air to catch flying insects. Or you might need a really big net if you're going to go and do some sampling in water, which you can do, as long as you hold the net in for a set amount, the same amount of time each time, sweep it around the same amount of time and use a net with the same size, then you can sample in water as well. Okay, so regardless of what piece of equipment you're using and where you're doing it, we normally use the mark, release, recapture method to estimate population size of animals. So you've got, first thing we need to do is we need to trap some animals or catch some animals in a net, and then you mark them in a harmless way. So what we mean by it is that it has to be non-toxic, so it can't literally, if they lick it or something, if they come into contact with it, it doesn't hurt them, and it can't prevent them from moving or anything to stop them getting food. And also it can't make them stand out to predators, because then they're going to get eaten, or they're more likely to get eaten. So anything that with strict movement to stop them feeding, or anything that's going to make them stand out to predators, is going to be unfair and non-ethical. So we need it to be something not dangerous. And then once you've marked them, you release them back into the wild, and then you come back again and either check traps that you've laid, or check the traps that you used before, or you sweep with the net again in the same place for the same length of time and everything a few days later. And then in your sample this time, you count the number of animals in total, and then you're counting specifically the ones that have marks on them, which are the ones that you would have caught before, marked, and put back into the environment. And so then we estimate this by looking at the proportion of the ones that you marked as a total of the whole population. So you do the number that you caught in the first sample times the number that you caught in the second sample, but you divide it by the number you caught in the second sample. And then that tells you roughly the percentage or the number, the population size of that organism in that area that you've done. Okay, so that was quite a quick tour, but we will just quickly have a look at what we have done and what we've looked at in this quick video. So we looked at why we must sample, and remember it's due to time and space. So physically, how many organisms there are, how large an area is, and the time it would take to count every single one. So we need to sample and then scale up. Reliable versus representative. So reliable, remember, means we need a large sample for it to be reliable, and representative must mean like the whole area, and it must be random. So using Quadrats to estimate the population size of plants or slow-moving organisms. So you are going to count the number in each quadrat and then use it to scale up for the whole area. And you could use this to look at the abundance of a species in different areas. So how many of a certain flower there are in somewhere that's been, the grass has been cut, and somewhere where the grass hasn't been cut, a shady area, a non-shady area. You can use this to directly compare different habitats or habitats that have been affected by different factors. You can use this, and remember, you're just looking at the population size or the number of organisms in each example. And then we looked at using Transect to look at the effect of a gradual change. So remember, this is looking at zonation, and remember, you're going to use a bar graph or kite diagram to show that. And it's showing gradual change in abiotic factor. So you need to go, your Transect goes across the habitat, and you're just losing one. What methods could we use to catch and count moving organisms? So we had said, so pooters could be one. We said pitfall traps, and we said nets of some kind. And then how do we estimate population size using mark and recapture? So you're going to mark, release, recapture them, and then use your formula to count how many. And that's looking for moving organisms. So when they can move, it's how you estimate their presence in a certain area, and then the population size of an animal that moves. All of that is a lot, but most of this is for sort of writing out methods questions, or thinking about answering data questions and analysis questions where they can give you data, or they can ask you to calculate a mean, or they can ask you to calculate a graph or estimate a population size given some data. So all of this is very useful for that. And if we didn't do these methods, we wouldn't be able to measure how factors affect biodiversity over time.
Ouch! This is why in some videos I have unexplained scratches.